The next speaker is critical to the development of nuclear medicine, particularly nuclear medicine therapy across the world. Dr. Hans-Georg Eichler is a regulatory authority. He's the senior medical officer in the European Medicines Agency in London. He's qualified as a professor and chair of the Department of Clinical Pharmacology, and he's also worked in industry with Ciba Geigy and Merck, so he's eminently well qualified to lead us through the regulatory labyrinth. Dr. Eichler.
Thank you very much, and thank you to the organizers for inviting me here. It will be a bit of a change of gear now. So here's my disclosure and disclaimer. I was asked to talk about the difficult development situations from a regulatory point of view. And I want to then talk about regulatory trade-offs and conclude with a few methodological considerations. So before we come to a difficult therapeutic situation or development situation, let me remind you of what the goal of a regulatory agency is. And in Europe, that is the EMA, or in the US, the FDA, and here in Australia, you have the TGA. So we look at quality, safety, and efficacy, or QSE. Therefore, any sponsor or company who wishes to bring a product to market has to show us their pharmaceutical data. This is about the product itself, preclinical or non-clinical studies, and most importantly, clinical studies. Now, we have only, it's a very simple job actually, if we reduce it to basics. What does a regulator look at? And what are the questions he has to look at? It's essentially two. Number one is, if you look at the totality of the data, do you come to the conclusion that the benefits outweigh the risks for a given described patient population? Everybody knows that. We're looking at benefit risk or benefit harm. By the way, we do not look explicitly at financial toxicity. Money is out of the equation when it comes to regulatory questions. It comes in later. That's a different ballgame. So everybody knows that question, and the answer has to be yes. But there is a second question that regulators have to address, and it's written here. All the estimates of the benefits and the harms are made under conditions of uncertainty. You've seen it just in the previous two talks. So what is it really? Is it that much or that much? So we also have to ask, what about the uncertainties around both the benefits and the harms? And the uncertainties have to be acceptably small. Now that is a judgment call because there is no hard rules for that. So it's those two questions. And our Canadian colleagues talk about BHU, benefits, harms, and uncertainty. It's very important because uncertainty plays a key role here. So much for the overall picture. Now let me introduce you to what we call increasingly the difficult development situation. And I'm starting with something that has nothing to do with your field of inquiry here. It's a totally different therapeutic area. Just to give you a glimpse. When I was a medical student, I learned about a disease called cystic fibrosis. And many of you know that if you have done medicine. Now that disease, pathologically, if you want, died away in 1989. Because then we detected that there is a certain gene. That gene expresses a certain protein, and there are different protein mutations. So we don't look at one cystic fibrosis population any longer, even though phenotypically they may be very similar. But as we move into a cause-based sort of treatment, addressing the wrong gene product, now that one population divides into subpopulation. And this is real life. We're already there. So for example, we had a product authorized, both in Europe and I suspect here as well, that addresses the gene product itself, the so-called cystic fibrosis transmembrane receptor. And we could ask the company to do randomized controlled trials for the subset of patients who had a relatively frequent mutation. But we know that there are other subsets that are so rare that even if you did a global study, you would not get enough patients to do an adequately powered RCT. So we had to make do for these less frequent mutations with accepting what no regulator likes to see. series, N of 1 studies, uncontrolled series. So the worst of the worst in our way of thinking. But has become a necessity. So that is one example. I'll give you another example that we will likely see in future. And that is about the combinatorial complexity of personalized treatments. I don't think anybody still believes that the future, certainly for cancer, is in single drug therapies. It will be combinations. The better we understand the pathology, the more obvious it is becoming that you need to hit the target over the head, or the cell in that case, not with one hammer, but at different sites with different hammers. And it's also becoming apparently, not only in cancer, but increasingly in other therapeutic areas, that you will have to do that individualized. In other words, if all of us in this room had the same quote-unquote cancer, say colon cancer, whatever you want, but we did a genome analysis on all these tumors, we would find that one of us needed treatments A, C, and F, and the next person here needed B, D, and Q, or whatever the case may be. So what that gives rise to, and we are approaching that situation, is what some people call the combinatorial complexity. And there was a very interesting paper that is quoted here, so I recommend reading if you are in this field. They said, look, assume we had a library of 40 actionable mutations, which we almost have now, and we only want to select combinations of 3 treatments. Then there are at least around 10,000 possible combinations. That is basic algebra. Anybody can do that. But then they said, look, with certain assumptions on the frequency of these various mutations, you might need to screen 25,000 patients to find 200 who have the same 3 selected mutations that you're interested in. That is only an exercise to illustrate the difficulty of real world that we're facing now. No one can do that. So we will, in all likelihood, not see RCTs for these complex situations. So that was another example. There are a few more of the so-called difficult development situation. But now let me come to something that is closer to your heart. Sorry, I forgot that slide. I think it's important. In the past, with evidence-based medicine, the research question has always been, is A better than B in a group of defined patients, where A is usually the experimental, the new treatment, and B is either a placebo or a proven active. Now, in future, because of what I try to describe in brevity, maybe the research question should be different. And it should be, if A truly modulates target X, i.e. it has been shown to have pharmacodynamic activity, how can we identify patients who benefit or combinations that work? It's a very different research question that will require different way of evidence generation. So now let me come to something that is close to your heart. I asked my colleagues in oncology when I came here, I said, what's our experience at the European Medicines Agency with theranostics? And the short answer he gave me was zilch. We haven't had a single application for what you call the PRRTs. So I said, well, anything similar? He said, yeah, here, this. So this is, I put it in bracket, almost a theranostic. I know it isn't in your sense of the definition. But there is also an entity for the diagnosis based on a radiotracer. In this case, it was to target a cell surface folate receptor in ovarian cancer, and it was coupled to a technetium-99. And then the therapeutic entity was not a radioactive, but it was a chemical, but it was still meant to be cytotoxic. I'm not telling you the story because this one is particularly important. But it's a cautionary tale, like so many we see where I sit. So there was a strong rationale. The logic of the whole approach was obvious. Yeah, why not do this? It must work. There were promising case series. We heard that we had treated 50 or 100 patients, and this was the median survival and all of that. And it looked very good. And we were almost on the verge of saying yes, when the results came in from a randomized controlled trial. and the efficacy was 0. So the company withdrew, this is dead. And I'm telling you this cautionary story because I'm always wary when I hear case series. We have treated X hundred patients, and again, median survival is this. And the question is, yeah, very good. But what is the counterfactual? What would have happened to those patients if they had not received this, if they had received a placebo or nothing or a different treatment? That is what we call the counterfactual. And I think we have to learn from these stories that what we think is logical isn't necessarily biological, because Mother Nature sometimes thinks differently. So that is my cautionary tale. And there are many, many, many such logical treatments that are obvious with a good rationale. Once you put them to the test, they disappear. Now, that's one side. But I'm aware that there is another side. And there are trade-offs to be made. And this is about who are we working for, how fast can we authorize treatments, the evidence versus access trade-off. So, to do that, please allow me to introduce you to two people. And I trust you have met both of them in one way or another. So, first, please meet Jane. Jane is in her late 50s, and she was recently diagnosed with an advanced cancer. She tried this and that, didn't really work. And she Googles and finds that her life expectancy is at max 2 years. Not a nice situation. So one day, Jane learns from someone, hey, there is a promising experimental treatment out there. Someone is trying that. It's still early days. The first results look good, but there's no convincing evidence yet. So what would Jane's reaction be logically? What would your reaction be if you were in her situation? I want to try that. I understand there is uncertainty, but the only certainty I have is what will happen without the therapy. That I know for sure. So I'm willing to accept uncertainty, and it's all about uncertainty. That's one person. Now meet John. John is also in his late 50s. He's in very good health. He's very intelligent and well-read. He knows he has a family history of cancer, so he knows that one day, very likely, he's going to get his cancer diagnosis. Like many of us, I'm one of these people. But you think, okay, well, that's probably 20 years out. It's not happening tomorrow. So when John hears, say at a polite dinner conversation, there is a promising treatment out there for this or that cancer. It's still early days, but it looks promising. John would probably say, good. I want more evidence on this. I'm not in a hurry. By the time my day comes, I want that there is sufficient knowledge about this so that the uncertainty is reduced to a minimum. I want to know what I let myself in for. Now, who is right or wrong, Jane or John? Up to you. I would put a question to you. Should our healthcare systems, and when I say healthcare systems, I mean the regulators, so my own camp, but also those payers we talked about, those who will have to pay for the expensive treatments, physicians, etc. Should our healthcare systems cater for the needs of A, only Jane, or B, John, or both? Can I ask you people for a show of hands? Who thinks the right answer to this is A? We should only look at Jane, so access is more important than evidence. Who thinks so? Raise your hands. No shows. One show. Okay. Who thinks it should only be John? That is the hardline regulatory stance. We will not compromise on evidence, whatever long time it takes. That's answer B. Who's here for answer B? No? One. Again, not a strong showing. Okay, last option. C. Should we cater for Jane and John to the extent we can? Please raise your hands. Thank you very much. You have just cast an almost unanimous vote in favor of what I call an iterative or lifespan approach to learning and on-market access. So, this is Elias known as adaptive pathways, big debate currently in Europe, to some extent in the US as well. So, why are we needing that? Why do we need this kind of adaptive learning and adaptive pathways to market? Because we realize that we have competing objectives. What I just showed you about Jane and John are two legitimate but competing objectives. And there is a tension here. So on the one hand, yes, we want to allow timely access for patients who are in need now, who say, well, I don't care what happens in 5 years. In 5 years, I'm dead. It's about now. Legitimate. On the other hand, we have an obligation towards society to only allow well-studied drugs with good evidence on the market. So how can we address that tension between the two? We want to enable precision medicine, and there are many other examples that I didn't show, those difficult indications that I just showed you. But at the same time, we know that we have good, reliable methods, good, reliable, objective endpoints. Unfortunately, they take a long time and they need a lot of patience and they're not always doable. Another tension here. And lastly, and I will not speak to that in this forum because this is more for payers, we want to ensure sustainability of the innovation engine, but at the same time ensure sustainability of our healthcare systems. And that brings us back to the financial toxicity. So I will park that last one. So, coming back to the idea of adaptive pathways, why this could be important also for the treatments that you are discussing here. What is a definition here? We start from the point that learning is a continuum. Learning doesn't end somewhere at a magic moment. So we want to plan the learning exercise prospectively. We want to take an adaptive approach to regulation and, dare I say, also reimbursement of drugs, because someone will have to pay for that. And if the payer, like here in Australia, you have a national payer, if that payer says, no, it's not on our list, then patients like Jane will not have it. So that's also important. And we say we will go through iterative phases of evidence gathering, followed by evaluation, and then by modification of the license or reimbursement. So if new information comes in, you change. You widen the license, you narrow it. In the worst case, you even take the product off the market. So we like to seek to maximize the positive impact of new drugs on public health for both Janes and the Johns of this world, by balancing timely access with the need to provide adequate, evolving information on benefits and harms. So the idea is that over time, we will come to that holy grail of very good evidence. Let me show that in a schematic way how the idea looks like. I ask you to please look at the top left-hand graphic. On the x-axis, you will always have time. On the y-axis, in case you cannot read the label, this is the number of patients who are exposed to a new treatment. So the dark red in the top left-hand means your classic development with randomized control trials. You have 3 steps, maybe you have 4, it doesn't matter. So you rely on RCT information. Patients are exposed in RCTs. And then comes this vertical line. This is when the drug hits the market. It's launched. And while you can still have post-launch randomized controlled trials, the vast majority of patients are now under that dark blue curve that grows rapidly because the drug is now up in the market. If it is taken up, the number of patients exposed will rise, and we will not learn normally from that patient experience. Those patients are treated somewhere in a hospital, in an office practice. Whatever happens to them has been lost to society or to decision makers. Only the yellow field, over the recent years, these yellow patients are meant to be patients in observational studies, disease registries, drug registries. So not randomized, but observational studies. What we want to do is change that dynamic, and I ask you now to look at the bottom left-hand graph. You could let the drug out earlier based on fewer studies, smaller number of patients. Yes, there is more uncertainty at the time of first launch. That is the left-hand initial license vertical line. But you recognize it, you acknowledge it, you inform patients. And then you do not stop developing. You continue, you can do RCTs in similar neighboring indications, but most importantly, you will gather the evidence, the information gleaned from every patient who is treated out there. That's why they are all in the yellow field. You want to get the patient experience, to contribute to the next round of evaluation. What does that mean? There is a blurring between the experimental setting where everything goes by declaration of Helsinki, informed consent, etc., and the next day where you have the clinical routine. Yes, you deliberately blur that distinction because you recognize that learning is a continuum. So you want the patient's number to grow slower, no off-label use, no uncontrolled use, but documented use and bring it back. So that's in a nutshell the idea. So what we will transition in future, and some people don't like the idea, but I think it's inevitable that it will come for many therapies. We move away from a magic moment where we say the drug was authorized on Monday evening, and on Tuesday morning we pretend that we know everything about it. Wrong. It's a lifespan management. We continue to learn. In the past, we were forced to predict what will happen in the market because we couldn't monitor. That changes. We can now monitor what happens. So we don't have to rely on prediction alone. And then, that is probably very controversial with some methodologists, certainly in my camp. The RCT is still the best methodology we have. But in future, we cannot rely on it alone. We need to embrace a full toolkit for evidence generation. And if that means observational studies, so be it. Because of precision medicine, we go from a focus from big populations to small populations. And because of that financial toxicity issue, the focus on licensing that we had 10 years ago will probably shift to a focus on patient access because a license without a reimbursement is meaningless. And something that will not ring well in the ears of some of my physician colleagues is we will have to probably move from open utilization, meaning that every physician can prescribe as they please. We will have to move towards more managed utilization. Whether it's the regulator who manages that or probably the payer who manages is a different matter. But the free-for-all is probably not a good idea to keep. So let me conclude with a few methodological considerations. If you have trained as a physician, as I have, a couple of years ago, well, several years ago, you would have learned under the evidence-based medicine mantra that the plural of anecdote is not evidence. We treated one patient and this happened is not much worse than saying we treated 1,000 patients and this or that happened. Always remember, you need to know the counterfactual. What would have happened to these patients if we had treated differently or not treated? That's why you got this hierarchy of evidence. And I'm sure most of you have seen this before. This is old news. Now, that was probably the best we could do 30, 40 years ago when this whole thing was invented. But can we make use of new opportunities that we have and apply them to the new challenges that we have? And the new challenges are these difficult indications that I spoke about. So let me start with something that is sort of a halfway and that even some of my statistician colleagues in regulatory science can accept. What has changed over the past 10 years? Two things. First of all, we now have data from past randomized controlled trials at a patient level. These are now available to us. They were not. There was a bit of fight over the past, I would say, 5 years. Now industry has given in. They make this available. There are now clearinghouses for data. The Wellcome Foundation is one, others as well. If you want to see patient-level data from an RCT that was concluded 3 years ago, you should be able to get that. We're on that trajectory. So in a couple of years from now, you say, I have a research question for which I need all the individual patient data from trials A, B, and C from the last 5 years, you will get that. Let me repeat that. We are on that way. The second new thing is that we have a rich data set of so-called real-world data from registries, e-health records, other types of collaboration, so outside the research setting. Can you make use of that information? So someone earlier in this session said the natural history of this disease is X and Y. So instead of putting a finger in the air and say, in an untreated cohort of patients with these and that characteristics, the median survival is X months, I guess, you could now prove that. You could now go back to data sets and say, yes, it is. So can we now borrow such data for RCTs? And people are now studying that, and these trials are now starting to be done, where you say we will not allocate 1-to-1, but we can allocate 3-to-1 and complement the control cohort with extant RCT data. This is work in progress, but it may be helpful for a situation that you are facing here. And lastly, could you go a step further? I didn't talk kindly about uncontrolled trials. I know that, and most of my colleagues wouldn't. But there is one sentence in a major document called International Collaboration of Harmonization. This is sort of a global document on the overall principles of drug regulation, where someone wrote in there a couple of years ago, it may be tempting in exceptional cases to initiate an externally controlled trial. Externally controlled trial is nice words for uncontrolled series, hoping for a convincingly dramatic effect. That's what we all hope for. When you started these treatments, you hope for a dramatic effect. Now, when it happens, good. Then you know that something is going on. This is an active treatment. But with a prompt switch to randomized trials, if this does not materialize. And given, unfortunately, the fact that Mother Nature isn't kind and doesn't always like our ideas, that latter half sentence will materialize very often. Because when you think it will be dramatic, well, it may not. So can you perhaps operationalize this? And a group of people have just published this. It's called threshold crossing. So you define an endpoint, overall survival, progression-free survival, whatever you want. You define your treatment-eligible population, basically your selection criteria. You define your estimand. I will not go into that. That's a very statistical term. And then you say, well, what is the counterfactual? What is the natural history if I treated those patients, say, with octreotide or with nothing? And with certain caveats, you determine the counterfactual from real-world data or past RCT data. Again, there have to be rules in place, etc. But can you do that prospectively and say, okay, we all agree that the median survival will be X months, plus minus. And then you set an efficacy threshold. Similar to what you do in a non-inferiority study. You say, okay, if this is my estimate of the counterfactual, then I will give it a markup to say if that new therapy is above that markup, the threshold, then I declare victory and say this is effective. And you have to agree that with different decision makers or the regulator, whoever else. And then only do you enroll patients in a single-arm study, and then you perform your sensitivity analysis. And if the threshold is crossed, as I say, you declare victory, you say, yeah, this is effective. If there is a very poor result, on the other hand, you also know it's over. You terminate the product. And if it's in between, you have now the situation where you can do an RCT because you are in a situation of equipoise. You have ruled out a dramatic effect. So then you can do an RCT, or if that is not possible, another single-arm study. So I think what we have to do in future for the difficult situations, come up with something that is better than the uncontrolled series, but that isn't where it isn't possible in RCT, so we can complement that methodology. Let me conclude. A lifespan approach to learning, licensing, reimbursement, etc. is needed to enable the translation of beneficial innovation to both current and future patients, the Janes and the Johns. As I said earlier, the dichotomy between research setting and clinical practice will be blurred and will need to be blurred. We will need a collaboration across and within decision-maker groups. When I say that, I mean first and foremost regulators and payers, because if these two do not march in sync, we will not achieve goal. I predict that evidence will be based on a more diverse family of data sources and methodologies that will complement but not replace the randomized controlled trial. I predict we will see a shift in focus from population to patients. And finally, a word to remember. Uncertainty cannot be eliminated. We will always have uncertainty around all the new treatments that we allow in the market. But what we try to seek with adaptive pathways is a progressive reduction of uncertainty and the hope that increasing the evidence standard over the entire product lifespan. Thank you.
Thank you very much for that very stimulating lecture. As physicians, we're all somewhat comfortable with uncertainty. It's refreshing to see the regulators are now becoming comfortable with uncertainty. The concept is coming towards the hospitality industry, I guess. Hotels for many years have had soft launches, so now we look forward to the soft launch of a drug. Professor Higgs?
Professor Eichler, thank you for that. That was an absolutely fantastic talk. Really enjoyed it very much. The challenge, I guess, that we face in neuroendocrine tumours, and particularly the theranostic paradigm, is that we're dealing with a disease that can arise from 14 different cells that has currently 3, probably 4 different grades of disease. And each of those sub-elements that we call neuroendocrine tumours is independently rare. So I'm sort of fascinated by your N of 1 or 2 type studies in this. Over 20 years, I've seen 3 patients with vipoma, for example, one of the varieties of NET, and I've treated them with PRRT, and they've all responded. So 100% efficacy in that particular subpopulation. But do you think I'll ever get a regulatory authority to approve PRRT in vipoma on the basis of those 3 anecdotal cases? And I'm sure there are other people in the audience who have 1 or 2 cases of vipoma. In your adaptive model, which is brilliant, I think, there is clearly, in academic centres, a will to continue to learn, but we need a partnership with government and payers to enable us to go on learning from our experience beyond the point of funding. And that's something that I think has been a failure. So I'd be interested in your idea of, particularly in this sort of scenario where you have a tumour that is very heterogeneous, that has very different behaviour, and the therapy is still being adapted as we go along. We're learning about it, of how you come to a funding agreement or model that enables that continual learning to occur.
So there was a number of questions. First you asked, I have 3 patients, can I get a license from a regulator? Well, 3 is a damned small number. No, just joking. But if you look, and people do that, there's a whole cottage industry of people who analyze what FDA or other regulators, EMA or here TGA, do. And if you look at those data that they publish, and they're true, you will find that we have authorized treatments on the basis of 15, 20 uncontrolled patients, uncontrolled case series. Nobody likes to do that, but we do recognize that there are such situations. And what you describe for the NETs is not so unusual. Pretty much all the really rare diseases that mostly affect children have those same characteristics. Very, very few patients, very high heterogeneity, and an unwillingness, even if there are more patients, to allow themselves to be randomized. And why is pretty obvious. So the difficulties you describe are not unique to your situation. So I guess you have to do what the best you can do. And the best you can do, if you want these therapies to be authorized, is to come up, collaborate with as large as possible a case series. But at the same time, and that is the advice that I would give to any company or academic, whoever is in that space, have information, credible information about the counterfactual. So if you come and say, I've treated 3 patients, they lived on average 2 years, that means nothing. If you come and say, I have 3 patients, they lived on average 2 years, but I have historically 200 patients from very different scenarios with very little variability, and they all lived never longer than 6 months, then the story may look different. It's because you can show, at least theoretically, even with bias and all that, you can hopefully show that there is a difference, and that's what we're after. So that was your first question. The second question is, how can you come to terms with the payer, sort of a contract, so as to learn and then revise? Now, in Europe we are having a big debate, and I know that here in Australia the TGA and what's the payers, the HTA body, the PBS, pharmaceutical benefits. Exactly, the PBS people, they're also having that debate. And it's pretty obvious. The one side says, the payers say, why should we pay a premium price for something that is essentially unproven? Reasonable position. Yeah, why should they? So, what is the way forward? On the other hand, not to pay for it, as I said, isn't the solution either. So maybe the solution is, and payers are now coming to grips with that, though reluctantly, because it's very cumbersome for them, and I do understand that. Can we say we pay for a certain period of time? Maybe we want an uncertainty discount from the company. That is a new word that is now coming into the conversation, and that is legitimate. And if in 2 years' time it turns out that your product isn't as glorious as we thought, which is very frequent, then we will either get money back or we stop reimbursement, etc. We will revisit. Now, there are many problems with that, but I can assure you that the discussion on this is now ongoing.
If there are no more questions, I'd like to... there's one more.
I would like to hear if you could comment on, it seems to me that it's very rarely that you see that rules in society gets loosened. That doesn't really only apply to this thing, but I mean, if you think about it, when we were kids, probably at a kindergarten, you could climb to a tree. Now it's probably deemed too dangerous in most places, especially here in Australia, I guess. But I mean, it seems like there is some kind of inherent mechanism in regulation that often things get tightened, but very rarely they actually get loosened. But it does have some effect on the overall ability of the society to have some kind of freedom. And I mean, Pasteur actually did cure rabies by being totally off the chart. I mean, he inoculated a patient without any authorization. And I'm not saying that we should do that, but I just think there is some kind of inherent mechanism that makes things almost always tighter, but very rarely more loose.
Well, that is a very philosophical question. I tend to agree with you, actually. But what we are realizing is that... So let me first say that I'm not standing here to say we want to loosen drug regulation. There is an accusation that this whole concept that I introduced to you is there to lower the evidence barriers. And my answer is absolutely not. We don't want to lower the evidence barriers because we would do a disservice to patients in the end. No one has an interest in having ineffective or toxic treatments on the market. And these barriers, or call them evidence standards, have evolved for a reason. So, no, we don't want to lower those. We also don't want to get higher. But we do understand that novel technologies cannot be approved and developed according to those strict rules that we had at the time when all the rage was antihypertensives and proton pump inhibitors, where you could do studies of 5,000 people relatively easily. So times have changed. We recognize that. So our conundrum is, exactly as you say, not to loosen. the regulatory standards, but at the same time enable that kind of innovation and not stifle it. And I would be lying if I wouldn't admit that we are still struggling with that. That is not an easy hatching.
Thank you. I think we're running a bit out of time, so thank you very much for the stimulating talk.